Welcome everybody to our weekly seminar, and I'm just here to open it and hand over to Ahmed to introduce our guest speaker today.
Hi everyone. Just shortly, our speaker today is Alba. She is coming visiting us from University of Lausanne, where she has been studying ants, social behavior, how individual processes relate to social behavior, and previously she did her PhD in Tübingen in Germany, studying something completely different from ants, great apes, and doing something with the stones that I didn't really get, but yeah, looking forward for your talk.
Okay. Yeah, thanks, Ahmed. So hi everyone. It's really nice to be here. Thank you so much for the invitation. So yeah, I thought today I'll tell you a little bit about the work that I've been doing over the last 2 years, looking at drivers of individual behavioral diversity, using ants as model systems. So today I'll tell you about 2 main projects that I've been leading during my postdoc in Lausanne. In the first one, I'll tell you a bit about an in situ investigation that we did of individual worker behavioral variation. So just as a little bit of background on the social life of ants, ants present division of labor. This means that different individuals in the colony are in charge of different tasks. The main level of division of labor is between the reproductive caste and the non-reproductive caste. In this case here, you have a social network of an ant colony, and there's a single reproductive individual, which is the queen in this case. Now, among non-reproductive individuals, there's also division of labor. This means, again, that they do different jobs in the colony. And here you have them colored differently. So in green, you have intranest workers, which are the nurses who take care of the brood and the queen inside of the nest. And then in blue, you have the foragers, who are the individuals who go out of the nest, find food, and bring it back for the others. And they're also in charge of other tasks, such as, for example, colony defense. Now, it is well established that in ants, the main task that an ant does in the colony generally predicts its sensitivity and responses to task-associated stimuli. However, there's not a perfect one-to-one relationship between main task specialization and behavioral profile, and this is due to 2 main reasons. The first reason is that ants can often and do often change tasks throughout the day. This means that at one point in time they can be foraging, but then at the next they can enter the nest and start cleaning. And the second problem with this one-to-one relationship is that the task that an ant performs in the colony is not always obviously functional. In some species, up to 40% of ants inside of the nest at any particular time are called lazy ants, and these are ants that are not doing any particular task at all. They're just kind of idle inside of the nest. So this means that this one-to-one relationship between main task and behavior is not perfect, which begs the question of whether responses to task-associated stimuli in ants are really modulated by task specialization, by the main task that an ant does in the colony, or rather by the task performance, so the task that an ant is doing at a particular point in time. Now, addressing this question is complicated for several reasons, both practical reasons and theoretical reasons. The practical reasons are that it's really hard to study task performance because classical ethological studies with ants mean that in order to investigate the responses of ants towards a stimuli, you take the ant outside of the nest, you put it in a colony, in an arena, you present the stimulus, and you measure the behavior. This, to start with, means that you're stopping the behavior that the ant was doing in the nest and causing a disturbance in the ant's natural behavior, because ants in isolation behave quite different from ants within the colony. The second problem is that if you try to take the opposite approach and you say, okay, we're going to do the experiments inside of the colony, this is quite complicated because, as you can imagine, when you have hundreds of individuals interacting with each other, it's pretty difficult to provide standardized control stimuli to targeted ants without disturbing overall colony activity. A theoretical problem with answering this question of differencing between task specialization and task performance effects on behavior is that task engagement actually is influenced by ant density. So ants evaluate conspecific density via the number of interactions that they encounter. And this has been shown to influence both task engagement and behavior. So for example, if an ant is not doing any particular task in the colony, but it starts interacting with a lot of ants that are, for example, cleaning, the probability that that ant starts cleaning is much higher than that ant starts doing foraging, for example. So the number of interactions an individual has with conspecifics, as well as the task those conspecifics are performing, influences task and behavior. These theoretical and practical problems led us to investigate the question of, can we differentiate the effects of task performance and conspecific density in modulating individual responses to stimuli, providing the stimulus in situ inside of the colony? Now, to answer this question, as you saw, there were some practical problems with answering this kind of question, so we had to develop a new method to address this question, avoiding or addressing these limitations. And to do that, what we did is we designed a new system that combines a robotic platform with an automated tracking system. And this, what allowed us was to provide standardized controlled stimuli to ants inside of the colony without disturbing overall colony activity, but at the same time tracking individual behavior and space use. So here you have a schematic representation of how the robotic platform looked like. And it's quite simple. It's composed of two main elements. One is the robotic dummy, or what we call the golden dummy, which you have here amplified. This just measures 2.5 millimeters, so it's almost the same size as a really small ant. And it's really just a magnet covered in gold with two little antennas, which are just rods to simulate an ant antenna, so we can provide tactile stimulation with this. And then this dummy is placed on an arena. This is where the ants live. This is the nest. And then this dummy is controlled by a series of rotary stages underneath that we can use to move the magnet around the nest, as well as to produce an oscillatory movement, because we can alternate current between these two rods here. And we can make then the dummy move at a frequency that we determine and during a period of time that we want. This means that we can move this dummy without disturbing all the ants inside of the nest, and we can provide a standardized control stimuli to the targeted ants that we want to investigate the behavior of. And for this project, we used Leptothorax athervorum ants, which are alpine ants that live in about 100 individual colonies in high altitudes in Switzerland. And as I mentioned, we paired this robotic system, or this platform, with an automated tracking system, which looks basically like this. Here you have, again, the arena where the ants live. This is the nest. This is the foraging area where they can go find food. You had the rotary stages here. This is the camera that we used to follow these QR codes that you saw in the previous picture, which are unique identifiers that allow us to monitor each ant's individual identity. And with this system, we are able to record the XY coordinates of each ant 8 times per second, meaning that we have a pretty good representation of a space use and individual behavior. In addition, this tracking system is placed inside of climate-controlled chambers, which means that we can reproduce more or less the climate that these ants would have in their normal habitat. We can control light cycles, wind speed, humidity, and temperature. Okay, so for this experiment, to address these contributions of task performance and density on ant behavior, we used 3 colonies of Leptothorax ants. Each of the experiments that we conducted with each of the colonies lasted 6 days. We performed 2 sessions per day. Each session alternated active hours with inactive hours. And what this means is that in active hours, we manually guided the dummy inside of the colony, like a video game. And we provided targeted stimulations to specific ants. And each stimulation consisted of 15 seconds at 1 Hertz oscillation. And we targeted 1 ant every 5 minutes during these active periods. During inactive periods, we just parked the dummy in a corner of the nest. Because we were interested in the effects of density in behavior, we also mapped the XY coordinates of all the ants every 10 seconds, meaning that we could build density maps to measure Basically, the color of the pixel in each of these maps means corresponds to the normalized density of conspecifics that we had around the ants that we were targeting. So this is the dummy, and if we would target this ant, we would just look at the pixel where that ant is placed. From each of these targeted stimulations, which looked like this, this is the dummy. You can see that it's more or less the same size as the ants, and it's going to interact with this ant. From each of these stimulations, we measured the behavioral response of the ants, the conspecific density around the ants stimulated, the brood density, but I won't talk about that today, and the task, of course, that the ant was performing at the time of the stimulation. So this is what we found regarding what types of behaviors the ants expressed towards the dummy. We found that they could respond in 5 different ways. They could be completely indifferent to the stimulation. This means that we couldn't see any obvious change in their behavior as a consequence of the stimulation. They could start antennating the dummy. This means that they would use their antenna to explore the dummy. This is mainly how ants gather information about their environment. They could perform excited runs. This means that once we interacted with the dummy, with the dummy, the ants accelerated considerably its running speed and started interacting with other ants that also started running, and this forms like a death vortex for like 2 seconds, and then the ants forget while they were running about, and then it ends very abruptly. But it's very clear to see, it's just all ants start running very quickly around the stimulus. Then we saw that the ants also climbed on top of the dummy, and we also saw biting behavior. So they would bite on these rods that we glued on the front of the dummy, as well as try to pull the QR code from the top of the dummy. And yeah, they just overall attacked it. Here you have the frequencies of each of these behaviors. So you can see that some of the behaviors we saw that happened alone, such as, for example, antennation or climbing on top, but there were other behaviors that only happened in combination, such as, for example, biting and excited runs. They only happened in combination with antennation as part of a sequence. So we had single behavioral responses as well as sequential behavioral responses. In total, we interacted with 208 ants, and we provided 551 stimulations, meaning that some ants were interacted with repeatedly. Regarding the analysis, we did quite a bit more than this, but I'm just going to present you these results today because they are the most relevant for the question at hand. The first analysis that we did was an investigation of whether ant density as well as task being performed at the time of a stimulation influence the responsiveness of the ants towards the stimulus. This means whether the ant responded in any way towards the stimulation. And then the second analysis that I'll present you today is again an investigation of the effects of ant density and task performance on the alarm probability of the ants. This means whether the ants responded with excited runs, attacking the dummy, or both. And this is what we found. We found that there was a negative relationship between worker density and probability of responding to the stimulation. This means that the higher the density of workers, the lower the probability of the ants to respond to the stimulation. And we also found that there was an effect of task on the probability of responding to the dummy, meaning that when the ants were doing brood care or were idle inside of the nest, idle means that they were not moving, but they had, when they were brood carrying or immobile, this means that they were either taking care of the brood or inside of the nest, not moving without antennal movement. This is like a sleep-like state. They were less likely to respond to the stimulation than when they were idle. This is when they were inside of the nest, not doing anything, but with antennal movement. So they were clearly awake. So yeah, we found also an effect of task on response probability. Regarding alarm, we again found an effect of worker density on alarm probability. The more individuals that were around the ants that we targeted, the lower the probability of alarm. But in this case, we didn't find an effect of task on alarm probability. An alarm was this combination of excited runs, biting, or both. Now, we thought that perhaps an explanation for our results of density-dependent behavior was that the density was affecting the ants' behavior via the number of interactions that the ants were experiencing in different density areas. So we tested this hypothesis of that ants perhaps in higher density areas were interacting more with conspecifics, and this was lowering their response thresholds, elevating the response thresholds towards the stimuli. So they were getting desensitized towards tactile stimulation. So to test this hypothesis, what we did is that we measured the number of interactions that each stimulated ant had in the 1 minute, 2 minutes, and 5 minutes before the stimulation. And we related this to, again, the response probability and their alarm probability. And we found that indeed this was the case for responsiveness. So the more interactions that an ant had in the 1 minute, 2 minutes, or even 5 minutes before the stimulation, the lower the probability of responding to the stimulation of the dummy. But this had no effect on the alarm probability. And this means that it was not the number of interactions in higher density areas or when there was more density of conspecifics that influenced the alarm probability of the ants, but it was something else related with density, such as, for example, perhaps the concentration of pheromones in the air. But we couldn't measure this, so this is just a potential hypothesis. Okay, so just to conclude this part, we found that task and worker density have independent effects on ants' behavior because the interactions between these two variables were not significant, but they have significant independent effects. We found that responsiveness and alarm are both density dependent, and that there seems to be a response habituation to tactile stimulation in higher density areas for the ants. But we found that alarm is not really influenced by the number of interactions, so it must be something else related with higher density of conspecifics that is influencing the behavior of the ants. Could be pheromones in the air, could be some sense of the way they measure safety in numbers, because if you're surrounded with conspecifics, the probability that there's some foreign individual there is lower, perhaps. We don't know that yet. Then we found that the probability of response is also context dependent. This means that it varied according to task, with ants doing brood care being the least responsive. We also found a difference between immobile and idle ants, so ants in a sleep-like state compared to ants that were not doing any particular task at the moment of stimulation. And we think this might have to do with task switching costs. So brood caring is a very costly behavior, and perhaps switching to interacting with the dummy might be too costly for the ants to do within the time frame that we measured the behaviors. Okay, so that is the first study I wanted to tell you about today. The second study I want to tell you about is how individuals as well as collectives respond to a disturbance in the system, namely an injury. And specifically, I'm going to tell you about a study that we did with Camponatus vela, which are carpenter ants from Florida. I bet you all know that predation is an important selective pressure that often leads to an arms race between prey and predators. Predation is obviously really costly for prey, but it can also be very energy consuming for predators, especially if the prey defends itself, because it can cause injury to the predators. And this prey defense can come in different flavors, such as, for example, morphological or anatomical adaptations, chemical adaptations, but also behavioral adaptations. And what you have here is an example of a Megaponera analis ant. This is a raiding ant that goes into termite mounds and steals the eggs and larvae to consume them in their nest. And what you can see here is a termite worker attacking the ant. And this is a great example that has actually led the ants to develop kind of defenses against the defenses. Because what the ants have done is that they have developed a system to ensure that these kind of injuries do not cost the life of many individuals in the colony. And they do this by performing two pretty unique behaviors. One is rescuing of injured individuals, and the other one is wound caring, or taking care of the injuries. This wound care this rescuing behavior is pretty unique. So the ants that have been injured after being in the termite mound actually signal to the conspecifics that they're injured by adopting a specific sitting position, as well as their cuticular carbohydrons change. So the other ants can smell that they have been injured, and then they come, pick them up, and bring it inside of the nest. And then other conspecifics, well, actually, we don't know in these species who are the ones providing care, but then there's other ants inside of the nest that take care of these wounds by licking them, as you can see here in this picture. And previous experiments done by one of my collaborators in this project found that if you focus here on the red line and the green line, the red line is the probability of survival of ants that have received an injury in one leg and did not receive treatment by conspecifics, whereas the green line is the probability of survival of individuals that have received an injury, but that they were allowed to receive social care for an hour by conspecifics. And you can see that there's basically an 80% difference in survival if the ants received wound care. So clearly it's efficient, it's adaptive that the ants are taking care of these injuries because it can really save the ant's life. So despite these advancements, in investigating rescuing behavior and wound care behavior in these termite raiding ants, there's still a lot of open questions in this system. We only know about this behavior since 2018. So in this project, we wanted to investigate whether there is a wound care package. Is there just one behavior that ants performed in order to take care of these injuries, or is it a combination of behaviors? We also wanted to know how does wound care impact the social environment of the injured ants. And we were interested also in finding out which are the ants that performed wound care, with the ultimate question in mind of are they doctors among ants. And we did these experiments using, as I mentioned, Campanatus vela as a model system, who is not a raiding ant, by the way. So basically, a master student that was in Lausanne for a couple of years found out that even though this species is not a raiding ant, they also perform wound care. So because we had them at the university, we used them as a system for this experiment. We used 3 colonies of Campanatus vela ants of 100 workers each, mixing, of course, nurses and foragers. And this is a bit the structure that the experiment had. So for the first 6 days, we track their behavior using the tracking system that I presented to you in the first experiment. This means that we could collect behavioral data as well as social network data during a baseline period. And then during the following 5 days, we did experimental manipulations. And for this experiment, we not only had 1 tracking system per colony, but we actually had 2 tracking systems paired and connected by a tube. And this is because in one of the tracking systems, we simulated nest conditions, so indoor dark conditions, whereas in the other, we simulated outdoor conditions. So like a foraging arena, as if the ants would go out of the nest and find food outside. And this would also ensure that the ants that were in the foraging box were most likely foragers, which was important for this experiment because every day of the 5 days of experiment, we took a pair of foragers from the foraging arena, and then to one of those ants, we cut we made an injury on the 3rd leg pair, and on the other ant, we didn't cut on the leg, but we manipulated the ant in the same way to make sure that it was not just the manipulation of the ant that was triggering differences in behavior from the conspecifics. Once we finished this manipulation, we put the pair back into the colony and we observed the behavior of the other ants towards these ants for the next 6 hours. And we did this 5 days in a row, so we had 5 pairs per colony, 15 pairs in total. And we found that there were 3 main behaviors that the ants did towards these injured and control ants. There was leg cleaning behavior, trophallaxis, and grooming. Leg cleaning, basically here in blue, you have the injured ant. This ant in yellow is taking care of the leg that we injured, but this other ant is taking care of another leg. So for this ant, we call this targeted leg cleaning or wound care, and this is just normal leg cleaning. Trophallaxis is the mouth-to-mouth exchange of food that ants do regularly to transfer food throughout the colony. And then grooming is just cleaning of the overall body of the ants that ants engage in for a lot of their time. So these are the 3 behaviors that we looked at for each of these ants during 6 hours post-treatment. And this is a bit of a zoom up of how the wound care behavior looks like. You can see here the injury, and you can see that the other ants basically start licking the wound, and it's pretty clear to see from the videos. And going on to the results, we found pretty clear differences in behavior between the injured and control ants. So here you have the control ants in blue and the injured in red, and you can see that cleaning of the target leg, which is again the right leg from the third pair, was much more frequent in injured ants compared to control ants, but cleaning of overall legs was equally frequent in both types of ants. So it's not that those ants were just overall cleaned more. Trophylaxis was also more frequent in the injured ants compared to the control, and body grooming, so not lead grooming, but overall body grooming, was also more frequent in the injured compared to the control legs. We also did this for the duration of the behavior, and we found the same pattern. The next thing we looked at was whether the network properties of these two groups of ants changed as a consequence of the treatment, and we focused on three particular network properties, namely betweenness, degree, and strength. Betweenness is the number of shortest path lengths that pass between any pair of nodes. The number of shortest paths between any pair of nodes that pass through an individual. The degree is the number of connections of that individual, and the strength is the sum weight of connections that an individual has. And of course, in order to tell that this treatment that we had done had any effect on network properties, we had to make sure that the network position and network characteristics of these ants within the colony did not differ before the experiment started. And this is what you can see here. This is in the 5 days before the experiment, this baseline period. And this is even in just the 6 hours before the experiment. And you can see that there are no differences between the control and the injured ants in any of these variables, in any of the time periods. We also looked at social maturity, which is a measure of foraginess. So it's a measure of the strength of belonging of an ant to the foraging community. And this was important in case, a nurse had gone into the foraging box and we had picked it up thinking that it was a forager, but in reality it was not. And because ants from different castes really differ in a lot of factors, it was important that we made sure that they were from the same caste. And indeed they were. The social maturity scores did not differ. So the degree of belonging to the forager community was not different between the two groups. Okay, so moving on to the results on network properties, we found that as a consequence of the injury, injured ants' betweenness decreased compared to control ants. You can see here the red line again is per hour. In each of the 6 hours after the treatment, the betweenness of the injured ants compared to the control ants. So the betweenness of the injured ants decreased. The degree did not change, so the number of individuals that they interacted with did not change, but their strength increased in the injured ants compared to the control ants. So to put it in other words, injured ants became more peripheral as a consequence of the injury than control ants, and while the number of ants they interacted with did not change, they became more connected to the ants they did interact with. The next question we wanted to ask was, who does wound care in this context? And for this, we basically, together with Evie George, who is my colleague in this project, we developed a system or a classification in order to make sure that we compared whether the ants that provided care with ants that had the opportunity to provide care because they encountered the injured ants, but they did not provide care. So we had to make sure that we compared ants that both had encountered the injured ant, but one group had provided care and the other group had not provided care, because we were interested in seeing if those wound care providers had any particular characteristics. To do this, we classified an encounter as an active movement from an away zone to an encounter zone. And an away zone was any area that was more than 1,000 pixels away from the targeted ant, whereas an encounter zone was a radius of 300 pixels, which is equivalent to 1 body length around the injured ant. So if one ant moved from this zone to this zone, we considered that this had been an encounter. And applying these rules, we found overall 46 caregivers. These were ants that had encountered the injured ant and had provided some sort of care. And then the non-caregivers were overall 66 ants, which were ants with similar encounter rates with the injured ants that had not provided care. So they had an opportunity for it and they did not. And then we compared two main characteristics of these ants. The first was the home range overlap of these ants with the injured ant before the experiments, and as well as the number of interactions of these ants with the injured ant also before the experiment. And what you can see here is that both during the hours pre-experiment and during the experiment, the caregivers had a significantly higher home range overlap with the injured ants than the non-caregivers, and they had a significantly higher number of interactions than the non-caregivers with the injured ants. So they were different. So in conclusion, in this study, we found that injured ants receive a special treatment compared to control ants, and injured ants become more peripheral and receive more attention from a fixed number of individuals compared to control ants again, and that wound-carrying ants share space and interacted more before the treatment with injured ants than ants that could have provided care but did not. And overall, I hope that from these two projects, I've convinced you that these new technologies are allowing us to investigate individual behavior in a social context and to analyze collective behavior with unprecedented resolution. From the first study, I hope you have learned that individual ant behavior is, at least in some cases, context and density dependent, and that from the second experiment, injury elicits not only specific behaviors from conspecifics, but also changes the social environment of the injured individuals. And hopefully from the last analysis that we're still doing more steps into, that wound care doesn't seem to be random and is performed by individuals that have specific relationships with the injured ants. And with that, I'd like to thank all my collaborators in both of these projects, as well as our universities and funders, and all of you for your attention.
Thank you, Alba, so much for this wonderful talk. Very interesting and inspiring to see the ants in this context. Are there any questions from the audience? And let's just pass around the microphone, and also the online audience can pose questions, and I will read them out if necessary.
Yeah, thanks a lot for the talk. One question about the social network graphs. What were the edges based on?
The edges? So you mean how do we derive the interactions between the ants?
Exactly.
Yeah, so around each of the ants, we built, I wish I had a graph of that. We built two ellipses. We built an ellipse around the head and an ellipse around the body. And then we considered an interaction when the head ellipses touch. And then that is considered an interaction, which is then what we use to do the social network analysis.
Okay, thanks a lot.
Thanks for the really nice talk. It's really cool to see this technology that you can use to look at the interactions really in place. I'm wondering about if you've thought about the role of the chemicals on the antenna, which could be, which in other species of ants is shown to, well, it differs from the brood workers versus the foragers. And this would definitely affect how the response to the interaction with the artificial ant. I was just wondering if you've thought about this or tried some experiments of changing the chemicals that would be able to identify this on the robot interacting.
Yeah, so great question. So the reason we first initially, the reason at the end why we coated the magnet in gold is not so it looks shiny, it's because supposedly we were advised that this would reduce the reactivity of the dummy to the smells of the colony, so it would be harder that anything sticks to it. Initially, we actually did these experiments either putting the dummy inside for a week or putting it fresh. And we thought that if perhaps they would respond differently, whether the dummy had acquired the colony's odor or if it was basically odor-free because we had put it in a chamber and then put it immediately into the colony. And we didn't find any differences between the responses to the fresh dummies compared to the dummies that had been there for a week. So I can't say exactly what they were smelling because the chemical analysis on the surface odors of the magnet were not very conclusive, and we didn't do it, for example, by putting the smell of a foreign colony. But at least from being for a week inside of the nest and a fresh dummy, there was no difference. And for each of the colonies, we used, of course, fresh dummies. So there was no cross-colony a smell that could be transmitted. But I agree there's a lot that we could do with putting different types of smells and changing the chemical profile of the dummy to see how they respond to this. It's in the future pipeline of altering the smell.
Cool, thanks.
May I follow up on this? So how do you consider this dummy? Is it an intruder or is it a nestmate?
It's a stone that moves. So like we didn't like for us it's not meant to be an ant. It's really just meant to be something that moves. We also compared, that's also why we had the inactive periods, because we wanted to see how the ants responded to the dummy when it was a dummy initiated interaction, so the thing was moving towards them, versus when the ant encountered this thing static. And we found that there were some differences. So they were much more aggressive, for example, if the thing moved, than if it was like static, which made sense. But for us, it's not meant to be an ant. It just has antennas because we needed some way to touch the ant and not ram into it. Because we got pretty good at the like video game interface, but we didn't want it to like throw away the ants. So that's why we had the two rods, so we could nicely touch them rather than send them away. But for us, it's not an ant. There were some thoughts of actually printing 3D and ant cases that we could put on top to like simulate different things, but we didn't find any printer that was small enough and detailed enough to give us a good enough resolution. But no, we don't perceive it as an ant. It's a moving stone.
Hi. So thanks for a nice talk. So my question is like on the wound caring part. So like how do you exactly choose which ant to like make it wounded? Like is it from one of the task doing groups or is it an external ant that you choose?
So because we had split here, the ants had basically two chambers where they could be, a foraging chamber and a nest chamber. And the idea was that in the wild, and for the species that are raiders, it's always the foragers that get injured. So we thought for ecological relevance, although these ants are not raiders, let's take foragers, because if anybody's gonna get wounded, it's gonna be the ants that leave the nest. So we picked two foragers at random, but this was really any two ants that were in the foraging box at a particular point in time. So it was not that they were doing anything in particular.
So like another, just one more question on this is like, do you specifically injure from a specific leg or
yes?
Like, because I see that you are cutting the backside leg, so basically there cannot be any kind of self-caring.
Yes.
So like,
yeah. So that was precisely because of that, because we wanted to control for the fact that they could not reach it at least easily, and because we had to choose a leg. So, but also in the wild. So these injuries are by no means fatal in principle. So of course they could get infected and then they could die, but they are by no means fatal. And ants in the wild can have up to 3 legs injured and continue raiding. So like, it doesn't bother them in the sense that they don't start walking like wobbly or anything. And I think that's probably, and when you see them, it's because they have still the other 5, which gives them a pretty good still stability. So if you would cut, I think, the front leg, they would kind of stumble forward. And that would make it so awkward for them to walk that they would just look completely different, probably.
Thank you very much for the nice presentation. And I probably it's a dumb question, but still, I'm very unfamiliar with the system. But why do you think ants, I mean, among all species, I would think that ants are the last one to heal someone else of the same species, because they are thousands, they are all sister, most of them steriles. And actually, I don't know if it happened also in the same species, but if there are some of them which are not even active, a percentage of them which is not even active, I would think that the easiest way would be just to replace the injured. Why, in your opinion, they do healing actually?
Yeah, we don't know. It's also because if you think here on the also like levels of selection for ants particularly, because the colony is kind of the most important unit of selection, and as long as the queen survives, you know, everything's fine. I agree, it might be cheaper to just let the, so that's the thing, I don't know what's cheaper, if it's cheaper to let the ant die or to provide 1 hour or a couple of minutes of wound care and save an individual. I'm sure it has to do with some cost benefit calculation that we are not sure of. I don't know why they do it. So if you also have to think about this in the sense that it's not, we don't think about it of like, a selfish thing that they go and do for their nestmates. Like that's not really the framework that we're using. It's more of, I bet there has to do something with, it smells in a particular way, a wound, especially if it's infected, then this leads to a behavioral response in other ants that then go and perform this behavioral pattern. It's very decentralized in that sense. There's not a lot of, I think, I don't want to say agency because they clearly have agency. It's not, I don't really know. But I agree, it must be some cost-benefit trade-off where it is cheaper to take care of the wound than to let the ant die and raise another ant.
Thanks, that's really, really cool. So one of the most interesting things about Leptothorax aceborum is that the colony itself has these profoundly strong rhythmic oscillations and activity. And I've always thought it would be great to use a little robot like yours to see if you can trigger these oscillations or get the oscillations to change phase. And did you ever try anything like that?
We haven't tried. We had planned an experiment for this, but we didn't get the grant. Sadness.
Oh, that's unfortunate.
But we had planned on trying to try to see if we could shift by basically activating the ants when they have these like low periods of activity, because basically they have these like peaks of activity and low activity levels throughout the day. So that's also why we run this like 1 hour on, 1 hour off cycles to make sure that at least at some point we caught both types of cycles. We had it planned that we would try to, in our mind, it was more like, can we shift from the less active to the active times?
Yeah, sure. No, that's also
we never did
super cool. It's just because these waves are typically activated in the most dense region and then spread remarkably isotropically across the colony, even with there's relatively few workers. And then the other thing that I thought would be really fun to try is with a species like Myrmica riginotus, or any of the myrmicas actually, that use stridulation or vibratory cues. So when they recruit, for example, for group recruitment, the recruiter goes into the nest and then just vibrates at a certain frequency, which I guess you could record and then put into your robot.
Yeah.
And the problem with existing work is that it's not clear to which degree that vibrational recruitment operates versus chemical recruitment, because you can't decouple them, but with your robot you could.
Yeah, that's a great idea. I mean, honestly, there was a lot of ideas. We also had, so there's also some species, for example, that had parasitoid queens that look a lot like their queens and just enter the nest and then basically take over. And we were thinking of building these models that we could then put on the robot and build a fake parasitoid and see whether they respond to it. There was a lot.
Again, Myrmica would be a wonderful species because they have these types of queens.
Yes, exactly. Yeah. So, I mean, there were a lot of potential extensions from this. This was really the first experiment we did with the system, but I agree there's a lot of possibilities.
So like I just have one more question. Like in your setup, is it possible that you can have more than one robot that you can control separately in different ways?
Yes. Yes, but we would need a larger setup, because I think in this case the robots would get pulled by the same, like the coupling is not strong enough that it would prevent it from attracting the other magnets. But if you would build a maxi version of this, you could probably have several stages doing several dummies at the same time. We also, so this was a lot of the PhD of Matthias, he was an engineer at EPFL, who did his PhD building this system basically. And he even managed at the end to make a closed loop circuit where basically he could just tell the dummy, go to ant 017, and the dummy would optimize the path to travel through the nest and go to that ant, avoiding the other ants. But then he finished his PhD.
Sounds good, yeah, thanks.
And left us for industry. So.
Thanks so much for the talk. That was super interesting. I have a somewhat simpler question just about the methods. So you had automatic tracking, so you could get the position identity of all the ants. But for the behaviors, were those manually coded or were those automated? Oh, okay.
Very manually coded. a lot of manual coding. We tried to use machine learning to do some kind of automated classification, and it just worked really bad. Like at the end, it was harder to clean the false negatives and false positives than actually to just do it ourselves. So we tried, but for example, brood care, it just doesn't, many of the behaviors doesn't work very well. And we tried to also use, work together with the people from Deep Lab Cut to get post-estimation and just infer behavior from that. But as soon as you have 3 ants on top of each other, which they do because they're ants, then it's basically a meta ant, and then the algorithm got completely mad. So,
thank you.
You do? Good. Yeah, maybe I should use your algorithm instead. But yeah, we did try to automate it because none of us wanted to watch all these hours. But at the end, we did. You also learn a lot by watching them, which it's a small price for the amount of hours you spend in front of it.
Thanks for the great talk. I was wondering if you could say a little bit about the evolution of the wound care behavior. So you made, of course it's difficult to answer that, but you made the point that the species that you're studying in the lab are not colony raiders, and that it was thought that this was a response to colony raiding in some kind of arms race.
Yeah.
And I was just wondering, do other ant species do this, or
yes,
just these two? Or,
they do. Yeah. Eric, actually, who started all this line of research, I just kind of jumped into it because I really wanted to, and then he let me do this. He got a grant to really explore the evolution of wound care behavior, and he's at a university in Germany that I'll tell you the name afterwards because I have forgotten. But he got a 5-year grant to study how phylogenetically wide this is, and what could be the triggers of why is it present in so many different species that have really so many different foraging modes. So exciton ants, I think, also have it completely different ecologically from some of these ants. He's looking into this, really trying to map how spread it is. They looked in the lab, basically we had, at some point, I think about 60 different species of ants, and then they looked, I think, at 10 or something, and they found it in 9. So I think it's way more spread than we thought, and it's really not only present in raiding ants, clearly. But he's looking at that. I'll tell you the name of the university later. But yeah.
I was just wondering, so for the experiments you showed at the end, looking at the care of the injured ants, do you just run, of course, as you quantify pre-experiment and then during the experiment, but do you ever repeat the experiments multiple times and see if, for example, that the ants, you have identity of the ants, correct? That the ants that could provide care and did, that they consistently provide care, or if it switches, yeah.
Great question. Yes, the answer is yes. And we looked at that, and there's some ants that, if you remember, we had 5 pairs because we took a pair every day over 5 days. This was a fault design because this means that the first pair that we put back then was back in the colony for 5 days. The last pair was back in the colony for 24 hours. So, bad experimental design. But we didn't think about it. It's only when we started seeing that actually the same ants that did wound care in some days did repeatedly wound care in the next days. And then we looked at whether we could find any differences between the repeated caretakers compared to the single caretakers and no caretakers. And we didn't find any difference, which made us think that maybe it was this opportunity problem that we were missing, that the ones that did not provide caretaker care just never met the ant. So that's where then we started looking at, okay, so we need to focus on those ants that encountered the injured to be able to tell something about the caretakers. But yes, indeed, there were ants that repeatedly gave care. We had some individuals that gave care over 5 days. So they gave care every single day. Are they any different from the others? No. But yeah, we did look at repeatability of wound caring, and we found that some ants gave opportunistic 1 day, and then some ants really gave on multiple days.
Really interesting. Thank you.
Thanks. So apparently there are no questions from the online audience. So calling last chance in the room. But if everything is answered. Let's thank Alba again for the wonderful talk.
Thank you.
And we close today's seminar. Come please back again next time, next week, same place, same time, and have a wonderful evening. Thank you.